1.
Hi, welcome to today's AP Biology lesson. Today our lesson will review material in Unit 2. On Friday, April 3rd, an announcement was made providing details about the 2020 AP Biology exam. The 2020 exam will be administered on Monday, May 18th at 2 p.m. Eastern Standard Time. All exams will be taken by students at home. The exam will consist of 2 free response questions. The first is a long FRQ and will have a 25-minute time limit. The second question is a short FRQ and will have a 15-minute time limit. All review lessons will include practice with science skills, which are relevant with any content. We will also provide practice with free response questions in each lesson. My name is Allison Kate. I'm affiliated with both UC Berkeley and Redwood High School, and today I'm teaching you from my home in El Cerrito, California. So just a little bit more about the exam format in case you're just joining us for the first time. There will be 2 questions. The first question is a long free response question, and it will have you interpret and evaluate experimental results. You'll be given a 25-minute time limit with an additional 5 minutes to upload your response. This will compose 65% of your exam score. And just a word about the timing. If I were preparing my students for the traditional exams that we've had in the past, I would tell them to plan to spend about 20 to 22 minutes reading the question and preparing a response for a long free response question. So you can see you have a little bit more time than I would normally recommend. In this question, you will be given a scenario that will be accompanied by data in either a table or a graph, and you'll be asked to do 4 different things with this scenario. And this is where the task verbs that we've been talking about in all of our lessons come into play. So in this case, in the first part, you're going to be asked to describe and explain biological concepts, processes, or models. In the second part, you'll be asked to identify experimental design procedures. In the third part, you'll be asked to analyze data. And in the fourth part, you'll be asked to make and justify predictions. And you can see how those points are distributed in the far right column. For the second question, which is conceptual analysis, you'll be given a 15-minute time limit plus an additional 5 minutes to upload your response, and this will make up about 35% of your total exam score. Again, if I were preparing my students, this is a 4-point short FRQ, so I would tell them to plan to spend about 6 to 8 minutes reading the question carefully and then preparing the response. So I think you have lots of time to be able to answer this question thoroughly. Here you'll be given a scenario that will describe a biological phenomenon with a disruption. And you will be asked to do 4 different tasks. Each task is worth 1 point. So in the first part, you'll be asked to describe biological concepts or processes. You'll be asked to explain biological concepts or processes or describe characteristics that are represented visually. And this is a science skill to look at things that are represented visually, such as models or data tables or graphs or cladograms or other graphic representations of some type of a biological concept or process. In the third part, you'll be asked to predict the causes or effects of a change in a biological system, and then you'll be asked to justify those predictions. Just another reminder that because this year's exams are modified, I want you guys to know that you are still going to be able to earn the full amount of college credit that you would have earned in other years. And this is a statement from the University of California system. And I suspect that most colleges will have their own statements, but I'm just going to read you the part about the UC system recognizing the effort that students have already applied to these challenging courses, and will award UC credit for the 2020 AP exams completed with scores of 3, 4, or 5, which is consistent with previous years. So let's go over our warm-up. Our warm-up was given to you as last night's homework, and this was given to you by my colleague, Ms. Pfizer. Let's take a look at the question. It had 4 parts, and you can look at those task verbs. As always, you should have your pencil and paper handy if you would like to follow along with me now, or if you did this homework last night, have it out so you can compare your answer to these scoring guidelines. Okay, so yesterday, Ms. Pfizer went over a lot of information about macromolecules, specifically proteins and nucleic acids. And so this question really is based on the fact that you need to understand that enzymes are proteins. Okay, so the stems and fruits of pineapple plants contain a group of protein digestive enzymes collectively called bromelain, and often used as an anti-browning agent for fruits and vegetables. Fruits and vegetables brown when they are bruised during transport or sliced and exposed to air. This browning is controlled by enzymatic pathways that produce brown pigments. The browning fruits and vegetables reduce the nutritional value of food, so anti-browning agents such as bromelain are used. In part A, you are asked to identify the type of monomer of which the enzyme is composed. In part B, bromelain works by breaking the enzymes that cause browning into smaller molecules. So explain how the reaction that breaks up the enzyme occurs. Part C, the pH of a solution determines the charge of certain R groups. The pH of pineapple fruit ranges from 3.5 to 5.2. You are asked to predict the effect of the activity of bromelain if it is used in a product with pH 11. And then you're asked to provide reasoning to justify your prediction. So there are 4 separate tasks: and identify and explain, a predict, and then justify your prediction. Okay, so if you're just joining us, hopefully you are able to answer these questions as we go. So let's take a look at the scoring guidelines. The first part, remember I told you that you have to know that enzymes are protein. So what make up proteins? What are the monomers of proteins? And of course, they are amino acids. So your response needs to indicate that amino acids are the monomers of the enzyme. In part B, you need to be able to understand how that protein is broken down. And 2 days ago, I did a lesson that went over the types of reactions. One of them is hydrolysis, and hydrolysis uses water to break apart the bonds in larger polymers. In this case, we're going to break apart the peptide bonds that hold the amino acids together. You could have also said that this enzyme breaks the bonds between amino acids using water as a reactant. In part C, remember we're taking the enzyme out of its optimal pH. So these enzymes work at a low pH, an acidic pH of pH 3.5 to 5.2. If you take it out of that and try to have this reaction occur at pH 11, the activity will decrease. It's out of its optimal range. And in part D, you need to have the justification for your prediction that the activity of the bromelain enzymes will decrease. And that's because we know that with those amino acids, they have R groups, and those R groups can be affected by pH. It can change the charge of the R groups, which would make the folding of the protein enzyme different. So it's ultimately its shape would be changed, and anything that changes the shape of the enzyme can ultimately affect its function. So there was a lot in this, from the monomer component to the types of reactions that build and break down organic molecules, understanding the optimum range for pH activity in terms of pH, and then how that pH affects the enzyme's activity. So let's move on to today's lesson. Here's our overview. We are reviewing today, and we're going to be reviewing 3 topics in Unit 2. We're going to take a look at cellular organelles. We're going to look at cell structure and function, and we're going to look at cell size. Today, we're going to practice with the science skill, making a scientific claim. We're going to do this today by looking at practice-free response questions that combine the content of these 3 topics and this scientific making a scientific claim skill. The learning objectives for today's lesson, these are the LOs from the course and exam description. You should be able to describe the structure and or function of cellular components and organelles. You should be able to explain how subcellular components and organelles contribute to the function of the cell. You should be able to describe the structural features of cells that allow organisms to capture, store, and use energy. You should be able to explain the effect of surface area to volume ratios on the exchange of materials between cells and the environment. And you should be able to explain how specialized structures and strategies are used for the efficient exchange of molecules to the with and to the environment. So here's what you need to know. This is the content part of today's lesson. And I'm going to start with cellular organelles, but I am not going to do an exhaustive list of all of the organelles found in cells. I'm going to be talking about the ones that are specifically mentioned in the course and exam description. So these are the ones that we're going to focus on. And the first one I'm going to focus on is the ribosome. And if you look at that third bullet point, the reason that ribosomes are so important is they are found in all living things. Every cell has a ribosome, and this reflects its common ancestry. So if you look at my image on the lower right-hand side, I've got two types of cells. I've got eukaryotic cells and prokaryotic cells. Eukaryotic cells have a membrane-bound nucleus, and prokaryotic cells simply have a nucleoid region. But you can see that they both have ribosomes. The other difference between prokaryotic and eukaryotic cells is prokaryotic cells do not have any membrane-bound organelles, and that means that ribosomes are not considered a membrane-bound organelle. The other things that you should understand about ribosomes is they're composed of ribosomal RNA and protein. You can get really down into the nitty-gritty composition by talking about the types of proteins and the size of the subunits. But for right now, just understand that they are composed of ribosomal RNA and protein. And that ribosomes, their function is to make proteins. Ribosomes synthesize protein according to a messenger RNA sequence that is transcribed from the instructions found in DNA. And we're going to have a whole lesson on that later on. So right now, we just kind of want to know that ribosomes make proteins. They synthesize proteins. The next organelle I want to discuss is the endoplasmic reticulum. It's kind of a mouthful, so we often refer to it as just the ER, and it occurs in two forms, the so-called smooth and rough. The rough ER is associated with membrane-bound ribosomes. And so if you look at the image, you can see all of these smaller ribosomes are attached to this surface of the endoplasmic reticulum. One of the functions of the rough ER is it creates compartments within the cell. And if you take a look at this, there are spaces, they're called cisternae. And there's so what's going on in this space could be a completely different type of chemical reaction than what's going on in this space. So there are multiple ways to divide up the cell, which is very small, and you can increase the surface area of that cell by dividing it up or compartmentalizing it. When I talk about this with my students in my classroom, I ask them to envision an office with a number of cubicles, and maybe the worker in cubicle 1 is on the phone and talking to somebody on the phone. Maybe the worker in cubicle 2 is working on the computer, and the worker in cubicle 3 may be just writing something with pen and paper. They're all doing different tasks, but they are separated by those physical barriers. And that's exactly what the endoplasmic reticulum does. It also helps in transporting the products of the ribosomes protein production. So it will synthesize proteins, and then those proteins can actually be transported through the ER to other places in the cell. A note about the smooth ER, you can see a little bit, I'll show you another image later on, but you can see a piece of it over here, and the smooth ER lacks ribosome, so it's not involved in protein synthesis, but rather it's involved in lipid synthesis. And it also plays a role in detoxification of certain molecules. The next organelle that I want to talk about is the Golgi complex. It goes by various names, the Golgi apparatus, the Golgi complex, Golgi bodies. We're going to refer to it as the Golgi Complex, although some of my images do still call it the Golgi apparatus. It was named, here's a little fun fact for you, it was named after an Italian scientist named Camillo Golgi. And students often ask me that because it is such an odd name. So the Golgi Complex is made of membranes. It's flattened stacks of membranes. And it also has some vesicles. And one thing I want to point out to you is that it has two sides, the so-called cis face, and this is where all the incoming proteins that the Golgi apparatus may be transporting and modifying. And then it has the trans face, which is the shipping face. So once the materials, the proteins move through this system, they get packaged, they may get refolded, they get modified in some ways, then they are going to be shipped out in a vesicle format. And so oftentimes when we talk about the Golgi, we refer to it as kind of the packaging plant of the cell, the shipping, receiving and shipping part of the cell. We'll talk more about the functions of all of these in just a minute. The next one is the mitochondrion, and everybody knows it affectionately as the powerhouse of the cell, and that's because the mitochondria is responsible for producing ATP, which is the main energy form that cells use. Mitochondria have a double membrane, and in this image, it's really nice because it color codes them. So the outer membrane is shown here in red. The outside is this tan color, but this is the outer membrane. And then there is another membrane. It's an inner membrane, and it's highly folded. It's convoluted. And again, you might be seeing variations on a theme. When we fold up this inner membrane, it allows us to increase our surface area and compartmentalize what's going on inside of this organelle. So it's got an outer membrane and an inner membrane, and the space between them has a fancy scientific name. It's called the intermembrane space. And then this blue portion that you see here has another name. It is called the matrix. And in a upcoming slide, I'm going to talk about the chemical reactions that are involved in producing ATP that will occur both in the confines of the matrix, and then also some that will occur in the actual cristae, the inner folded membrane of the mitochondrion. The next one is the lysosomes, and lysosomes have the nickname of a suicide sac, and that's because they are responsible for getting rid of any things that have made their way into the cell that need to get broken down. Lysosomes are membrane-enclosed sacs that contain hydrolytic enzymes. We talked about these. Hydro means water, lytic means to bust apart. So hydrolytic enzymes are going to break down any foreign particles or worn out organelles or anything that shouldn't belong in the cell. Another thing that lysosomes do is perhaps the cell has engulfed some food and has it enclosed in a vacuole. The lysosome will fuse with the vacuole, unleash its hydrolytic enzymes, and digest that food vacuole inside the cell. And then the last one I'm going to talk about, oh, not the last one, there's one more after this, is the vacuole. And vacuoles are found in both plant and animal cells, and they have a variety of functions. Let's first look at the plant cell because they play a really important role in plant cells in maintaining the sugar pressure of that cell. You might remember that plants have a cell wall that surrounds them, and so this is an opportunity for the cell to store some water and maybe some solutes to increase the sugar pressure of that cell. The membrane that surrounds the large central vacuole in a plant cell is called a tonoplast. And I have one example of a vacuole in an animal cell, and I'm going to talk much more about this in later lectures when we talk about how organisms maintain their osmotic pressure and potential. But this is an image of an animal cell. This happens to be a paramecium, and parameciums are filled with macromolecules and solutes, so they're hypertonic to the freshwater environment they live in. And that means that water is going to move into their cell bodies. These contractile vacuoles act as pumps, and they pump out the water to maintain that water balance. And if they weren't there, this organism would drown because the water would be flowing in at a rapid rate. Other things that vacuoles do in animal cells is they store food and waste products until they can be either digested or removed from the cell. And the chloroplast. I believe this is our last organelle today. Chloroplasts are specialized organelles that are found in photosynthetic algae and plants. They are the sites of photosynthesis. Chloroplasts have a double outer membrane. And I'm going to go into more detail when I start talking about the function. And then, of course, we have a whole lesson on photosynthesis. So you'll have a lot of practice with this structure. But for right now, I want you to see a similarity with the mitochondria, which is the organelle that I already discussed. And it has, the chloroplast also has a smooth outer membrane. Further, it has a smooth intermembrane. It's got an intermembrane space. It's not labeled. Then it's got another compartment, and this compartment is the stroma, and it's a dense fluid-filled compartment where parts of the photosynthetic reactions occur. And then the chloroplast has a third membrane system, and this third membrane system is called the thylakoid membrane. And inside the thylakoid is yet another space, and it is called the thylakoid space. And that is where the light reactions, the light absorbing reactions of photosynthesis will take place. And over here, you can see chloroplasts are tiny. So we're looking at an actual slide of some plant cells. I believe this is from a water plant. And you can see all of those teeny tiny chloroplasts that are inside that plant cell. So I'm going to go into detail about the functions of each of these organelles, but these are kind of the statements that come from the CED, the course and exam description, about each one. So let's read them together, and then I'll go through them using images to support this content. So the endoplasmic reticulum provides mechanical support. It carries out protein synthesis on ribosomes that are attached to its membrane, and it plays a role in intracellular transport. The mitochondrial double membrane provides compartments for different metabolic reactions. Lysosomes contain hydrolytic enzymes, which are important in intracellular digestion and the recycling of cells' organic materials, and program cell death, which is called apoptosis. And vacuoles have many roles, including the storage and release of macromolecules and cellular waste products. In plants, it aids in the retention of water to maintain turgor pressure. So now we're going to look at each one of these organelles and their function. So I've got an image of the endoplasmic reticulum. On the left side, I've got the rough ER, and on the right side, I've got the smooth ER. And I want to focus on the rough ER. The rough ER, as you can see, has all of these ribosomes that are attached to it. So ribosomes synthesize proteins. So the ribosomes that are attached to the ER make proteins that are going to be exported from the cell. They will work together with the Golgi to actually modify, package, and transport these proteins out of the cell. Okay, so it does provide that transport function. It provides the work surface for the ribosomes, and it is connected to the Golgi, which will modify those proteins for export. Just to contrast that, can you see in this cell there's also some proteins that are free? They're not bound to the ER. Those ribosomes, these are ribosomes not bound to the ER. These free ribosomes are actually going to be making proteins that are for use in the cell itself. So there are two proteins that are, or I keep saying that, I'm sorry. There are two ribosomes that are free, and they make proteins that are going to be used within the cell. And then, of course, the ribosomes that are attached to the rough ER are going to make those proteins that are going to be exported from the cell. And then you can see the smooth ER, it lacks those ribosomes, and it is not involved in protein synthesis, but rather in lipid synthesis. So it can make fatty acids, it can make phospholipids, which are used in cell membrane construction, and also steroids. And its detoxification function, it will detoxify molecules that are found in alcohol, drugs, and metabolic waste products. This is the mitochondrion, and I promised you that I would show you the two different places where the reactions that produce ATP are located within the mitochondrion. And again, you're going to have a whole lesson on cellular respiration in a later review session. So this is just to pinpoint where in the mitochondrion these reactions occur. So the Krebs cycle occurs within the matrix of the mitochondrion, and the electron transport chain, which is what really makes the lion's share of the ATP, is a series of proteins that are actually embedded in that inner membrane, in that highly folded membrane that we call the cristae. So again, we see that part of the production of ATP occurs in the Krebs cycle or the citric acid cycle. That's in the matrix, and the rest of it, ATP production, occurs along the inner mitochondrial membrane. And then we can follow up with the chloroplast because it has a very similar structure. And I know that Ms. Pfizer talked to you about the endosymbiotic theory. And now you can really get a look at why chloroplasts and mitochondria are once believed to have been their own free-living prokaryotic organisms that were engulfed by a cell. So these guys, these chloroplasts, with their membrane system, and if you look at this image, they have that outer membrane, that inner membrane, and a third membrane system called the thylakoid membrane. Thylakoid literally means stack of coins from the Greek, and so when they're stacked up, we call that stack a granum, and you can see within that chloroplast there are multiple grana. Surrounding the thylakoids is the stroma. Now, the light reactions, or where the pigment chlorophyll resides, is in the thylakoid membrane. So the first part of photosynthesis, or the so-called light-dependent reactions, occur within the thylakoid membranes, and the Calvin cycle, or Calvin-Benson cycle, which is where we actually fix our carbon dioxide into our organic carbohydrate molecules, our sugars, occurs in the stroma. So those two reactions take place in different parts of the chloroplast, just like we saw the different parts of cellular respiration reactions that make ATP occurring in different parts of the mitochondria. So I hope you're seeing the similarities between these two organelles. The last topic today is about cell size. And if you've ever wondered why cells are small, this is the lesson for you. So it has to do with the cell's surface to volume ratios. And it turns out that the larger the surface to volume ratio is, the more efficient the cell is going to be. And the cell needs to interact with its environment to obtain nutrients, to eliminate waste, to acquire or dissipate thermal energy, or otherwise exchange chemicals and energy with the environment. So this is a lab activity that you may have done while you were in AP Biology class. I know that Ms. Pfizer is going to go over this with you tomorrow in your review lesson, but this is just an image right now to show you that the extent of the diffusion of that white part of this cube is the greatest in the smallest cell, and that's because it has the largest surface area to volume ratio. Oh my gosh. So here's a bunch of mathematical equations and formulas, because when we look at surface area and volume, there is some math involved. And so these equations are lifted straight from your AP Biology equations and formula sheet. And I want, again, I want you to be familiar with these so that you can see how they're used in the AP Biology curriculum. So we're going to focus on the surface area of a sphere and the surface area of a volume, which are the first two equations. Let me again point out the key points, take-home points from the course and exam description. The surface area of the plasma membrane must be large enough to adequately exchange materials. And we're going to see in some later slides some ingenious ways that cells increase their surface area. These limitations can restrict cell size and shape. Smaller cells typically have a higher surface to area volume ratio, so they have a more efficient exchange of materials with the environment. As cells increase in volume, the relative surface area decreases and the demand for internal resources increases. So what you tend to see, instead of cells getting really, really big, is that we end up making more cells. And so we have multicellularity rather than one great big cell or one great big organism. More complex cellular structures like membrane folds are necessary to adequately exchange materials with the environment. I have several examples coming up. And as organisms increase in size, their surface area to volume ratio decreases, which affects properties like the rate of heat exchange with the environment. So you'll also see some organisms have specialized structures to dissipate that heat. Or in some cases, they have behavioral mechanisms to do thermoregulation. So here's some examples of some different strategies to obtain nutrients and eliminate waste that organisms have evolved. If you look over here, this is what your intestinal cells look like. So you know that you absorb all the nutrients from the food that you eat through your small intestine, and your intestine is lined with these projections, and these projections are called villi. And again, those folds increase the surface area so that you can absorb as many nutrients as possible. On top of villi, you have microvilli, which are even more extensions to further increase the surface area for nutrient absorption. If we look at gas exchange organs, we can see that there are some external gills and we have lungs. And so I want to focus in on the mammalian lungs because this is what your lungs look like. You can come down here and get an enlarged view of an air sac or an alveolus. Your lung is made up of millions of these small air sacs. And what happens when you get down to that level, we increase the surface area for diffusion of oxygen into your lungs and carbon dioxide out. So it greatly increases your surface area. And another fun fact for you, the surface area of your lungs for gas exchange of oxygen and carbon dioxide is roughly equal to the surface area of a tennis court. So that's kind of an amazing fun fact. And then one final thing about fish, and we'll talk about both freshwater fish and saltwater fish in a later lesson, but they kind of go to the extreme. So what happens is they have gills and they swim. This is a freshwater fish, I believe it's a trout, and it's swimming through the water, and its gills have set up something called a countercurrent flow, where the oxygen-rich water will flow into the oxygen-poor blood, whereas the carbon dioxide from the blood flows back out into the water. So they have really super efficient exchange through gills. So those are lots of strategies that different cells and different organisms have adapted to increase the efficiency of the exchange of nutrients and waste. So let's get some practice with these ideas. These are all going to be some former free response questions. This first one is from an exam that was given before 2012, and so it's an older question. So the scoring guidelines are a little bit different than the types of exam questions you'll see on your exam. But this question reads, during an investigation of a freshwater lake, an AP biology student discovers a previously unknown microscopic organism. Further study shows that the unicellular organism is eukaryotic. You are asked, your task verb is to identify 4 organelles that should be present in the eukaryotic organism, and then to describe the function of each organelle. Now I just want to also point out a test tip to you. When you are asked to identify 4, just identify 4. We will only score the first 4 that you identify. So you are wasting time by listing more than those 4. So just give us your 4 best ones. And then you're asked to describe the function of each organelle. So remember that this is eukaryotic. So think about the 4 that you want to talk about. I've given you more than 4 in this lesson. Grab that pen and paper and write them down. And then let's see if we can match the function to each one. Okay, so I know this chart is a little light. And let me tell you how this was scored. So you got 1 point just for identifying and listing your 4 organelles. Okay, and then for each of those organelles you listed, you would earn an additional point if you had the function correct. And the reason that these organelles are listed in the order they're listed is that's how often they appeared in the responses by students the year that this question was given. So because it was eukaryotic, a lot of students said nucleus, which I did not specifically talk about in this review, but the nucleus is found in the eukaryotic organisms, eukaryotic cells surrounded by a nuclear envelope, and it contains the hereditary information, the DNA, the chromosomes, or the genes. The next one listed is ribosomes. Ribosomes synthesize proteins. And then we have the ER, and you could have scored a point for just talking about generalized endoplasmic reticulum, or another point for talking about rough and or smooth ER. So we know that the endoplasmic reticulum is an internal transport organelle, and it compartmentalizes the cells. That when we specifically refer to the rough ER, it is important in protein synthesis, and the smooth ER is particularly important in lipid synthesis and also detoxification of certain substances. Mitochondria is responsible for producing ATP. Chloroplasts are responsible for photosynthesis, which produces carbohydrates using sun's energy, solar energy. Then we have vacuoles, vesicles. They are different, but the distinction is it's kind of a fine nuance. So they could be lumped together here, and they can be for storage or transport. We did not discuss cilia and flagella, which are used in cell motility. We did not discuss basal bodies, which are a support for cilia and flagella. Golgi bodies, which again are the packaging plant. They modify proteins, package and transport these modified proteins. Lysosomes, we mentioned, contain hydrolytic enzymes and break down any waste products, metabolites, or pathogens. And then an organelle we did not mention is a peroxisome, which is involved in detoxification of specific reactions that produce hydrogen peroxide, which is toxic to cells. So hopefully you were able to list at least 4 organelles and describe the function of each of those. The next one is going to ask you about the concept of protein production, modification, and export. So this question reads, a molecule of messenger RNA, which is abbreviated mRNA, has just been synthesized in the nucleus of a human cell. So that should give you a hint about what kind of a cell we're dealing with, i.e. eukaryotic. If the cell is a secretory cell, explain how the protein is eventually targeted, packaged, and secreted to the exterior of the cell. So since it's going to be secreted, we're going to talk about that the ribosome is likely attached to the endoplasmic reticulum, and we're going to explain how it gets targeted, packaged, and sent to the out of the cell. So this was worth 3 points, 3-point question. So any one of those bullet points would have earned you a point. First of all, there are special proteins called chaperone proteins that help fold that newly synthesized polypeptide into the protein, that it could be modified in the Golgi by the addition of sugars or phosphate groups to somehow get in the response that there is an endo or internal membrane system. In other words, that protein is moving from the ER to the Golgi, where it is packaged up into vesicles, and that those vesicles are moved to the surface, and then they fuse with the cell membrane, and that protein would be released to the outside through the process of exocytosis. One more practice. This is a question about cell size. You are given some data, which you're provided with the radius of 2 spherical structures that are surrounded by a membrane. And you were asked to calculate the volume and surface area of each of these. And again, the equations are given to you, and you're given the radius. So all you have to do is plug that in. So let's take a look. So to calculate the volume, you're going to use the volume of a sphere formula, which is the volume is equal to 4/3 times pi times the radius cubed. And the radius in the first cell is 1. That makes that math super easy. And the radius in the second case is 3. And your units are going to be millimeters cubed. And then to calculate the surface area, it's 4 times pi pi times r squared. And again, the first one is easy because it's 1. The second one is a little bit more challenging, but then you would get your answers. So in the first case, you have a volume of 1 cubic millimeter and 1 square millimeter for the surface area, which is a 1-to-1 surface area to volume ratio. In the next case, you're going to calculate the volume to be 23 millimeters cubed, and the surface area is 9 millimeters squared. So the surface area to volume ratio is 3-to-1. Remember, the higher the surface area to volume ratio, the more efficient the cell will be. Okay, so let's do a check for understanding by practicing with our skill using the content about cell size and this skill of making a claim. So I'm giving you a data table, one like you just saw, and it's got 3 cubes, a 2-centimeter side with a surface area of 24 square centimeters and a volume of 8 cubic centimeters. We've got an intermediate one that is 1 centimeter with a surface area of 6 and a volume of 1 with a 6-to-1 surface area to volume ratio. And finally, we have a small one with the sides are 0.5 centimeters, giving us 1.5 centimeter squared surface area and 0.123 cubic centimeters volume, which works out to be a 12-to-1 surface area to volume ratio. You are asked, using this data and the image below, to make a claim about cell size that could be supported using these observations and calculations. All right, you got that paper? Let's make a claim. What is your claim about cell size that you could use these data to support? So did you say smaller cells are more efficient at moving materials in and out of cells than larger cells? So how would you support that claim? So here's your evidence. The smallest cube had the least amount of that red pigment remaining. The largest cube had the most amount of red pigment remaining, and the medium cube had a medium amount in between the other cubes. The reasoning here is that it's clear when you observe these 3 cubes that the smallest cube would be the most efficient at moving materials in and out. The smallest cube had the largest surface area to volume ratio, 12-to-1, while the largest cube had the smallest, 3-to-1. And so it would be the least efficient. Okay, so that's a little practice with making a claim, which is to state, assert, or maintain that something is true. Keeping that claim in mind, you need to also think about the evidence you have to support the claim and the reasoning you have to connect that evidence to that claim. All right, so your learning objectives today. You should be able to describe the structure and or function of cellular components and organelles. You should be able to explain how subcellular components and organelles contribute to the function of the cell. You should be able to describe the structural features of cells that allow organisms to capture, store, and use energy. You should be able to explain the effect of surface area to volume ratios on the exchange of materials between cells and the environment. You should be able to explain how specialized structures and strategies are used for the efficient exchange of molecules to the environment. Your key takeaways, these are your enduring understandings, are that living systems are organized in a hierarchy of structural levels that interact, and that highly complex organization of living systems require constant input of energy and the exchange of macromolecules. All right, before I give you your homework, let's take a look at some common errors and misunderstandings. This was a AP exam question about cell structures. And then I want to share with you some global things that students did when they responded to this question that readers noticed when scoring it. So you're given a data table, and the data table has 3 types of cells: cell type X, cell type Y, cell type Z. And then you've got the relative amounts of organelles in the 3 cell types. So let's look at cell type X. It's got a small amount of ER, both rough and smooth. It's got a large amount of mitochondria, and it has cilia present and a small amount of Golgi bodies. Cell Y has a large amount of both types of ER, smooth and rough. It has a moderate number of mitochondria, no cilia, and a large amount of Golgi. And finally, cell Z, doesn't have any of those things. None of those organelles are present in cell Z. Your question: is based on the analysis of this data, identify a likely primary function of each cell type, and then explain how the data support the identification. So you're making a claim. You're making a claim that the function of cell type X is blank, and then you're using the data to support that claim. Okay, so let's take a look at how students answered this. So cell type X, because it had cilia and a large amount of mitochondria, the mitochondria produces the energy and the cilia move. They move things along surfaces or they move an organism. So the function of this cell type is likely some type of movement. Okay, don't name the cell. You don't need to name a muscle cell or any specific cell. You are identifying the function. Okay, cell Y had a lot of ER and Golgi, and so it's going to be involved in some type of protein synthesis and then secretion of that protein for export. If you focused in on the smooth ER, then you're going to talk about lipid synthesis. So either one of those, that you're not going to get points for both of those, but either one. And again, notice that we're not naming a specific cell type. We're identifying a function and we're supporting it based on what we saw in the data table. And cell Z. Cell Z could have had a number of functions. It could have been for transport. It could have been for, and let me give you an example because that one is kind of tough. When it says oxygen transport, so we know that red blood cells transport oxygen. So red blood cells don't have any other organelles other than that hemoglobin protein, which actually transports the oxygen. It could transport water in plants, and we'll see in a minute that the cells that do that are xylem cells, and they're dead. They have no living tissue, so they have no organelles present. They could be protection. You know, your outer epidermal cells are dead. They don't have any organelles. Support, like that xylem. Storage, like the adipose tissue, which has no organelles, stores fat. And then no function at all. The cell could be dead or is undergoing cell death, which is called apoptosis. So none of those require organelles. So let's see what the readers saw as common errors and misunderstandings. The first thing is that students did not make the connection between the function of the cell and the organelles present. Rather, they identified the special type of cell. And I tried to point that out to you when we talked about cell X. A lot of students just said it's a muscle cell because it needs lots of energy and it has mitochondria, instead of identifying the function of movement. Students often identified cell X as cellular respiration and didn't link the ATP production by the mitochondria, which is providing energy to the cilia for movement. Students were often confused about the function of organelles. They interchanged the functions of cilia with villi, and hopefully now you can see the difference. Cilia is involved in movement or transport along a cell surface, whereas villi increase the surface area of a cell. They often stated that the endoplasmic reticulum makes ribosomes and that the ribosomes get exported from the cell. So clearly the endoplasmic reticulum supports the ribosomes and they make the proteins that get exported from the cell. And finally, students had a lot of difficulty when presented with cell Z that lacked organelles. And I know when I gave this to my students, they were challenged by that as well. So you just have to remember that there are some cells that do not possess all of those organelles. Think red blood cells, think xylem, think adipose. And then often students named cell types like prokaryotes or stem cells or neurons, rather than identifying the function. And so it's really important to read the question carefully. All right. So let's take a look at your homework tonight. So your homework tonight is 2 organelles that we talked about today, mitochondria and chloroplast, and there's a long introduction, but what you're asked to do with this question is to describe the importance of inner membrane separating the different regions of both the mitochondria and the chloroplast. Okay, so I want you to read that question carefully. I want you to look at the diagrams that are presented visually for you, and then I want you to describe the importance of those inner membranes. Tomorrow, Ms. Pfizer will be with you, so she will debrief this homework. I'd like to thank you for watching and hope that you'll join us for all of our lessons. Be sure to complete the practice question because it'll get reviewed tomorrow. I will see you next week, so have a great lesson tomorrow and a great weekend. Thanks for watching.